In today's video, we shall learn about kingdom Fungi. By the end of this video, you will have a clear understanding of fungal mode of nutrition, degrees of parasitism, fungal structure, vegetative, asexual and sexual reproduction, and finally about the major groups of fungi. Let's start with the introduction to fungi. The branch of biology which deals with the study of fungi is called mycology. Fungi are non-green, that is, they lack chlorophyll and are nucleated thallophytes, means not differentiated into root, stem and leaves. Fungi may be unicellular or very complex multicellular organisms. Fungi possess a definite cell wall made up of complex polysaccharide, namely chitin. The reserve food material is in the form of glycogen. Let's look into the mode of nutrition. Since chlorophyll is absent in fungi, they obtain their nutrition from other sources. They are called heterotrophs and their mode of nutrition is heterotrophic nutrition. Fungi are classified into two categories. First, saprophytes, which obtain their food from decaying organic matter. And second, parasites, which live in or on the living organisms and obtain food from them. When the fungi grow on the external surface of the host, they are known as ectoparasites. When the fungi grow in the tissues of host plant, then they are referred to as endoparasites. Let's take up degrees of parasitism. Obligate parasites. They depend on a host for their survival, that is for nourishment, reproduction and other activities. Facultative saprophytes generally obtain their food from the living matter, that is, they are ordinarily parasitic but can obtain food even from dead organic matter, that is, they may be saprophytic. Facultative parasites, normally they are saprophytes but may behave as parasites. Obligate saprophytes are strictly saprophytes and cannot live a parasitic life. Symbiosis. Both partners are mutually benefited. Example, lichens, a fungus living symbiotically with an alga. Second example is mycorrhiza. Myco means fungi, rhiza means roots. A fungus grows in close association with the roots of higher plants. It may be ectotrophic or endotrophic. In the former, the fungus lives on the surface, and in the latter, the fungus penetrates into the root tissues. By the way, if you are getting some value out of this video, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now we shall look into the fungal structure. It is a thalloid structure, not differentiated into root, stem and leaves. It has a cell wall, plasma membrane, cytoplasm, nucleus, vacuoles and other organelles like mitochondria, ribosomes, endoplasmic reticulum, etc. The thallus or hyphae is collectively called the mycelium. Coming to kinds of mycelia, aseptate mycelium and septate mycelium. Aseptate mycelium, due to absence of partition or cross walls or septum, the hyphae becomes multinucleated and aseptate and is known as cenocytic. It is found in algal fungi Phycomycetes. Septate mycelium, due to presence of septa at regular intervals, the mycelium is known as septate mycelium. It may be uninucleate or multinucleate. It is seen in the members of Ascomycetes and Basidiomycetes. Next is reproduction. In fungi, reproduction is of three kinds. First, vegetative reproduction, which occurs by vegetative or somatic portion. Different types of vegetative reproduction are fragmentation. The fungal mycelium breaks into several fragments and each grows into a new individual, seen in Rhizospores. Budding, as the name suggests, a small bud-like structure is formed on the parent cell and gets detached to form a new individual, seen in yeast. Fission. The parent cell divides into two equal halves and each half develops into a new individual. Again, example is yeast. Sclerotia are resistant and perennating bodies. They form a pseudo-parenchymatous structure. They remain dormant during unfavorable conditions and germinate into new individuals during favorable conditions, seen in Claviceps. Rhizommorphs are pseudo-parenchymatous structures formed by interwoven hyphae. They carry on with the functions of roots. Like Sclerotia, they remain dormant during unfavorable conditions and grow during favorable conditions. Next is asexual reproduction. The different methods of asexual reproduction are: first, by zoospores. Zoospores, which are motile, flagellated spores formed within the sporangium, seen in Phytophthora. Sporangiospores are non-motile spores produced inside the sporangia. Example, Rhizopus. Chlamydospores. They are thick-walled, resting spores formed during unfavorable conditions and are rich in reserve food and are resistant to desiccation. With the return of favorable condition, they develop into new individuals. Oidia. The hyphae breaks into small pieces which act as spores. They are thin-walled and germinate immediately. Conidia. They are non-motile spores produced exogenously, singly or in chains by constrictions on conidiophores. Next is sexual reproduction. In fungi, sexual reproduction consists of following phases. First is plasmogamy. In plasmogamy, the two sex cells come close to each other. Fusion of both the protoplasm takes place. There is no fusion of the two nuclei and the cell becomes binucleate or dikaryon. Second phase is karyogamy. In karyogamy, fusion of both the nuclei takes place, resulting in the formation of a diploid nucleus or synkaryon. Syn means coming together or fusion, karyon means nucleus. Finally, this is followed by meiosis or reduction division, resulting in the formation of haploid nuclei. Kingdom fungi is divided into different classes depending on the morphology of the mycelium, modes of spore formation, and fruiting bodies. The major groups of fungi are Deuteromycetes or imperfect fungi, Phycomycetes or algal fungi, Zygomycetes or conjugation fungi, Ascomycetes or sac fungi, Basidiomycetes or club fungi. Phycomycetes and Zygomycetes belong to lower fungi, while Ascomycetes and Basidiomycetes belong to higher fungi. Let's take up Deuteromycetes. The mycelium is septate and branched. They are known as fungi imperfecti because sexual reproduction is either absent or not reported. Reproduction occurs only by asexual method by the fusion of conidia. They may be saprophytes or parasites or decomposers and help in cycling of minerals. Next is Phycomycetes or algal fungi. The mycelium is aseptate and cenocytic, means multinucleated. Asexual reproduction takes place by motile spores called zoospores or by non-motile spores called aplanospores. Spores are produced endogenously inside the sac-like sporangia. Sexual reproduction may be isogamous, that is, the gametes are similar, or oogamous, gametes are dissimilar, otherwise known as anisogamous. Zygospore is formed by fusion of two gametes. So plasmogamy is followed by karyogamy and meiosis. It is seen in Phytophthora infestans, which causes late blight of potato. Next group is the Zygomycetes, otherwise known as conjugation fungi. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by non-motile Sporangiospores inside the sporangia. Sexual reproduction takes place by copulation or conjugation between two identical gametangia. It results in the formation of diploid zygospore. During germination, the zygospore gives rise to a promycelium seen in Rhizoppus stolonifer, which causes soft rot of sweet potato. Next group is Ascomycetes or the sac fungi. Sac fungi are cenocytic, branched, saprophytic decomposers, parasitic or coprophilous, growing on dung. Asexual spores called conidia are produced exogenously on special hyphae called conidiophores. The conidia on germination produce mycelium seen in Aspergillus and Penicillium. Sexual spores, namely ascospores, are 8 in number, produced endogenously in sac-like asci. The fruiting bodies are known as ascocarps. The ascospores are liberated from ascus and germinate to form new mycelia. Example: Aspergillus flavus produces aflatoxins, which are carcinogenic. Second example, Claviceps purpureae causes ergot of rye. Finally, we come to Basidiomycetes or club fungi. The mycelium is branched and septate. Vegetative reproduction is by fragmentation. Plasmogamy takes place by the fusion of two vegetative cells of different strains. The dikaryotic structure gives rise to a club-shaped basidium. Karyogamy and meiosis takes place in these basidia, resulting in 4 haploid Basidiospores exogenously. In higher forms, the fruiting bodies are called basidiocarps, seen in Agaricus campestris and is known as gilt mushroom. So today we have learnt about different modes of nutrition, degrees of parasitism in fungi, various modes of reproduction, and also about the major groups of fungi. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.